Julia Kuharenko, and I'm a junior at UW, and I'm studying biochemistry.
My name is Christina Mukin, and I'm also a junior at UW, and I'm studying biomedical sciences. And the topic of our research was Sanger sequencing of fish DNA in order to discover mislabeling of fish in stores and distributors. And our research question was, is tuna most likely to be mislabeled because it is cheaper to source other fish similar to tuna?
Our hypothesis is that tuna will be the most likely to be mislabeled when compared to tilapia, imitation crab, true cod, mackerel, and bonito flakes. Our research and design methods were performing DNA barcoding of the cytochrome C oxidase subunit 1 gene on 2 different pieces of fish muscle. The gene is considered a marker which varies even in closely related species, so it could be used to identify fish to the species level. In order to obtain the DNA from the fish sample, DNA extraction was performed. After DNA extraction, the process of polymerase chain reaction is used to amplify the CO1 gene in order to make multiple copies of it. PCR uses the different temperatures in order to help DNA to denature, primers to anneal to single-stranded DNA, and for DNA synthesis to occur. Afterwards, gel electrophoresis is done to determine the quality of the DNA sample extracted. Sanger sequencing is then done to determine the nucleotide sequence of DNA. Lastly, bioinformatics are used in order to analyze the nucleotide sequence obtained by Sanger sequencing.
And our results were, after inputting the nucleotide sequence into forward and reverse barcode of life database, as well as forward and reverse basic local alignment search tool, we revealed that only 3 out of the 4 tests matched greater than 90% accuracy. However, the accuracy of the reverse barcode of life database sequences were less than 60% accurate. Therefore, this was not included in our final data. To further this research, the trials will be expanded to other cities and countries to determine the probability of mislabeled fish in specific regions. And in our conclusion, this research is important because mislabeled fish can pose a health risk to the consumers of the fish. For example, if the consumer has an allergy to a certain type of fish, or the fish imported from other countries contains chemicals that fish sourced from the US do not have. This research also applies to population studies because if fish aren't correctly labeled, then the correct populations will not be known. This can cause a decline in the fish species due to the population of mature fish that are able to reproduce falling below a population threshold. Thank you.